If I knew I had this fuchsia halo, I would have chosen a different color shirt. I think they brought the party equipment and not the conference speaker monitor. Was that you, sir? So, good afternoon. My name is Nick Conley. I am a principal scientist at Locus Biosciences. I joined Locus in July of 2018. They acquired my San Francisco Bay Area company that I co-founded called EpiBiome. I made the move across the country to the Raleigh Research Triangle Park area to lead Locus's process development, formulation development, and GMP manufacturing efforts of our CRISPR bacteriophages. So these are phages that have been engineered with a CRISPR Cas3 system, so that once they infect bacteria, not only do you get the wild-type lytic activity, but you also get that genome degradation. So we target that CRISPR to the genome, and the Cas3 very efficiently digests it. And these combined mechanisms of action result in much greater depth of kill. So I'm happy to say it was a very successful manufacturing campaign. We'll be dosing our first patients very early of next year. I'm not here representing Locus, and I'm not talking about bacteriophages. Instead, I'll be talking about GMP manufacturing considerations for viral vectors gene therapy. So I want to start at a very high level, and then we'll get down into the weeds. I'll introduce GMPs, talk a little bit about the IND Module 3, which is the Chemistry Manufacturing Control section. I'll talk about the FDA draft guidance for industry for human gene therapy for rare diseases. Talk about the precedent that exists for AAV9 gene therapy, although I don't think I have to do much of that because the last speaker did a great job. Talk about manufacturing of recombinant adeno-associated viral vectors, some differences between R&D scale manufacturing and scalable manufacturing, and then finally some of the critical quality attributes and release testing that will be required before clinical trials can begin. So, what are CGMPs? Stands for Current Good Manufacturing Practices. These are statutory, so they are actually written into federal law. That CFR stands for Code of Federal Regulations. The purpose is to provide for systems that really assure that the pharmaceutical drug manufacturer has very good control over both the processes and the environment or the facilities. This provides some confidence that the identity, the potency, and the purity of the drug that it's manufactured in a controlled manner. And when you don't follow them, there are really two ways to run afoul. One is misbranding. That's pretty straightforward. That's saying the label doesn't represent what's in the product, false or misleading. And the second is adulteration. Adulteration isn't how we think of adulteration. It's not like it's a contamination necessarily. The FDA doesn't actually have to prove your drug's adulterated. If you're not following GMPs, they can conclude that it is. And then there's all sorts of penalties. You can get in trouble. 483 observations, which requires management, at the CEO, director, quality level. There are warning letters. They can put your facility under consent decree, recall your product, take away your marketing approval. As we learned from Theranos, they can ban you from working in the industry. There are fines and even prison in extreme cases. So that's one reason to follow them. But there are actually a lot of other good reasons to follow them in terms of establishing the quality of your product. These are 10 principles. I won't go through them in detail. Not all of them apply at phase 1. I've actually put the ones in bold that do apply. But there are some, what we'll call phase appropriateness of GMPs. So, you won't be doing any process validation until phase 3. You don't need to do release testing on your raw materials at phase 1. And then for phase 1, interestingly, the VP of manufacturing operations can also serve as director of quality. Those two roles together. As you can imagine, there could be a conflict of interest if the person releasing your drug is also the one responsible for the output. So FDA separates those roles at phase 2. So module 3 is where you convince the FDA that you did manufacture your drug in a controlled manner. A lot of companies get tripped up here. There are a number of promising therapies and promising companies that failed because they got put on clinical hold because of an insufficient module 3, and they went on to run out of money and go out of business. As you can see in this this what's called the CTD. CTD is Common Technical Document. It's the standardization of the IND format from the US to the EU to Japan. You can see that Module 3 is really at the foundation because it is a critical aspect of the IND application. So this guidance, human gene therapy for rare diseases, it was developed because of the realization that, hey, there are pharmaceutical companies working on this, and rare diseases are rare. By definition, they affect less than 200,000 Americans, but they are not rare themselves. There are nearly 7,000 of them, and collectively they afflict more than 25 million Americans. Most of them are caused by a single gene defect. So this is a very large but highly segmented market. Biopharma has entered this space very aggressively, and you can see that from the precipitous drop-off in the small molecule programs. But genetic disease is complicated. These therapies are very complicated, and despite all the investment, the development of these therapies is really too slow to satisfy people that are afflicted by a rare disease. So having a guidance is a good step forward, and at least providing some knowledge. FDA recommended that there was a demand, recognized that there was a demand for this knowledge. So what the guidance says is that this is hard. So you've got small study populations. You're going to be doing fewer manufacturing runs. There's less opportunity to actually understand the manufacturing process. They go on to say that, however, demonstrating process control to ensure consistent product with the predefined critical quality attributes for potency, identity, and purity is required to demonstrate compliance with licensure and regulatory requirements. So if we translate that from FDA speak, what it means is saying, what you're doing is what you're doing is admirable, and we know that it's very difficult, but there are no shortcuts. So you're going to have to follow the same rules as everyone else. And, that's not a bad thing because there is a certain finality with gene therapy. We don't have good technology to reverse it today, but it does establish the expectations. And not only is there FDA guidance, I think for SLC 6A1, we're fortunate that there's precedent for a successful gene therapy for another neurological disease. That's the giant axonal neuropathy that you heard about in the last talk, and delivered through what is presumably the same route of administration, a lumbar puncture, and presumably in the same patient population, PEDS. So, you never want to wake up, find that, you've got a family member or a loved one that's afflicted with a rare genetic disease, but at least there's a guidance for industry now and precedent. And I included this reference. I don't know if you're going to circulate these slides, but this is the reference for the really nice preclinical work out of Dr. Gray's lab. So, I dug into that because I was very interested in the manufacturing of these viruses. It looks like for much of that work, it was the UNC Chapel Hill viral vector core method that was employed. This viral core was acquired by Bamboo Therapeutics. That was in January of 2016, and then Pfizer acquired Bamboo in August of 2016. But some nice work by Grieger, where in this review showed actually a lot of different gene therapies that are currently in the clinic. And what you'll recognize right away is that the methods are highly fragmented. So large differences in viral amplification, whether it's a transfection method or stable producer packaging cell lines, HeLa lines. There are even insect expression systems, baculovirus systems, and that's just to produce the AAV. The purification methods are also highly fragmented, so you could see that fourth column there, all sorts of different ways. And part of this is that it's just an immature field. There hasn't been convergence on an optimal way that works for all AAVs. But part of it is the proprietary nature of biopharma. So, the publications that you'll see are largely from academic institutions, some of which have an interest in doing GMP manufacturing, but many of which don't have a big interest in scaling. And so most of the information that's published is limited. Fortunately, Grieger did go to some length to actually generate a general method that is widely applicable. It begins with generating GMP plasmids and a master working cell banks, HEC293 cell basically culture and then transfection. Those cells are then harvested. They're lysed either mechanically through sonication or freeze-thaw cycles, or chemically with the use of a detergent or some osmotic gradient. That naked DNA is then digested, and then through a combination of usually density gradient centrifugation and chromatography, those empty capsids and those process impurities like host cell DNA and host cell protein are removed. And then finally, there's often a concentration and buffer exchange step, as sterile filtration and filling to put that into a product presentation that's suitable for parenteral delivery. So there's a nice reference there on that generalized method. I still think that there are a few different areas for improvement. And then I also wanted to highlight some of the differences between an R&D approach and a scalable GMP approach, kind of convolving the two. There are small-scale GMP approaches as well. But in an R&D approach, you might go out, say hi to your buddy and ask him for his last passage of HEC 293 cells. You may take your undergrad's maxi prep and use that to generate your AAV vectors. In a GMP manufacturing environment, this begins with a well-characterized GMP plasmid and master cell banks. Excuse me. The next sort of thing that happens in the R&D approach is, there's often use of tissue culture flasks, and even in manufacturing environments, there's an attempt to scale those. But ideally, you do something like adapt the HEC 293s, which are often generally adherent, and then adapt them to grow in suspension. And then you can use a stirred tank bioreactor, and then you're scaling volume instead of surface area, which is much more footprint friendly. In terms of clarification, you see a lot of centrifugation in academia. In industry, we use depth filters. Depth filters help deal with that cell debris bioburden. In academia, you'll see a lot of dialysis tubing. You might even see centrifugal molecular weight cutoff filters. In industry, you'll often see tangential flow filtration membranes, where you have a lateral flow that keeps the membrane from fouling, and then there's a pressure difference that drives things across the gradient. And then chromatography is largely the same, but you do have Part 11 compliance issues, which deal with data integrity. And the GE Act, it just doesn't, I don't believe that's an instrument that can be validated readily. In terms of manufacturing challenges, getting the titers really high, this takes a lot of material, which often requires very large volumes. Removing process impurities, there are actually some pretty tight specs for what's allowed in terms of host cell DNA in that final drug product. Removal of empty capsids, where you might have immunogenicity, but no therapeutic benefit from those empty capsules. That's currently done with density gradient centrifugation today, which is a very horrible, not very scalable method. And just in talking with Dr. Gray, I think that's one area for significant improvement. And I understand he's making some strides in that area. Obviously, minimizing purification losses, ensuring stability, and then Sorry, I got ahead. And then developing phase-appropriate analytical methods for proving identity, potency, and purity. So I just want to end with some of the release testing that almost certainly has to be done. Appearance testing is very standard, especially for parenterals. These are things like, what does the drug product look like? Colorless to slightly yellow, clear to opalescent, essentially free of visible particles would be a typical spec. It's very important to know the viral genome titer, that's PCR assay, as well as the infectious titer, and then you can take a ratio to get the ratio of full capsids to infectious particles. Identity is very important. You can do whole genome sequencing to get at what's inside of the capsid, and in fact any other impurities that come along. But you also need to test to make sure that you actually have the right AAV capsid that's encapsulating that DNA. Because this is a gene therapy, you need assays, functional assays, not just genomic assays, but you need to show that you've got the expression, the anticipated expression levels right, and the activity. You also need to show that you've removed almost all of the residual host cell protein, host cell DNA. If you added any sort of elements that are concerning, you may have to prove that those are not absent in the drug, that those are absent or substantially reduced in the drug product. Because it is a parenteral and it doesn't have the benefit of the protection from your GI system, almost certainly you will have to have bacterial endotoxins, particulate matter and injection, so a light scattering technique to show how many subvisible particles you have greater than or equal to 10 micron or greater than or equal to 25 micron, pH, container content for injections that prove that you actually filled the vials to the right volumes. Stability is essential if you're going into the CSF. And then finally, an ICH stability program. So you are required to show that you are drug product is stable even for phase 1 clinical trials. So it looks like a lot. It is a lot, but it is manageable. So these analytical methods, they can be developed pretty quickly. All of this can be outsourced. Unfortunately, there's not a lot of compromise, even though it's early stage, just because of the parenteral nature of how these things are delivered. So I'd love to take any questions. Thanks, Amber, for inviting me, giving me a last-minute speaking slot. And thanks to all of you for giving me a great lesson today. I learned a lot.
Oh, any questions? Oh, sorry. Jane first.
So I certainly take into that element from the manufacturing perspective that it's still relatively young and there's a maturation going on. But I do think that there is a little bit of difference than, say, monoclonal antibodies and other complex biologics, in that I think there's considerable data in the field that different AAV serotypes in terms of the manufacturing components are going to be different. And if you make it by different methods, that you actually get different products at the end. It's just really there's an additional layer of complexity that is such that you may not have the kind of homogeneity in terms of manufacturing that you may see with, say, monoclonal antibodies or something like that. Yeah.
Yeah, this is definitely a unique challenge, but I still think very addressable, and that's been demonstrated, I think, with commercial success and commercial limited number of commercial products.
On the technical side, for the full quantification or DNA quantification, where do you think things are with respect to qPCR versus digital droplet PCR? Isn't the last one sort of overtaking, becoming the standard?
Yeah. So you won't get pushback. I don't think you will get pushback. I was not involved in the IND filing on any of the GAN work. I think at phase 1, qPCR is probably fine. I'm really excited by digital droplet PCR. We have a large effort underway at Locus. I think, if you can get down to single molecule, well, I mean, I used to do single molecule spectroscopy. I don't like ensemble averaging. I think it is the future.
So in the process, the manufacturing process, what steps are actually covered as intellectual property? Something that you're basically protected from, say, generic insults almost immediately when you start to manufacture?
Yeah, that's a great question. I have a little bit of experience from that. I took some coursework at the Stanford Law School. And in general, you don't want to patent something if it's not discoverable. So if I can't go and look at my competitor's product and prove that he used my method, then patenting it will only disclose it, right? So it's possible there could be a whistleblower, maybe a disgruntled employee who might come over and tell me, but I don't want to have to rely on that. The things that I can probably patent more reliably, things that are discoverable, but things like a cell line, right? Since Myriad Supreme Court decision, you can't patent naturally occurring DNA. Interestingly, USPTO has issued several patents on things like natural viruses, including bacteriophages, with the requirements that they're significantly different from anything that's ever been reported, that they're sequenced, that they're deposited into a bank like ATCC. There's one in Canada that USPTO also accepts. I think the focus should be on things that are discoverable in the process, and it could be that if you use a certain compound, that the residuals could be discoverable, and that would provide insight that your competitors are, infringing. But that's how I like to think about it.
Okay, I think we can move forward. Thank you so much, Nick.